So we start with the first session of today, and we all know what happens in the gut does not remain in the gut. So the impact of microflora, inflammation, and dysbiosis of gut in behavior and symptoms of autism and how it can impact on the child and caregivers is huge. So we have Dr. DEEPAK Gupta to provide us an insight on the importance of gut and the interventions that can be included to improve autism, to improve the sign and symptoms of autism by especially emphasizing on microbiota stabilization. So Dr. Deepak Gupta is a well-known name when it comes to treatment in autism in India. He's the founder and director of Center for Children and Adolescent Wellbeing. Over the past few years, he has helped a lot of children with autism and related learning disorders to improve and recover. So over to you, sir, and a very warm welcome.
Yeah, good morning, everyone. Let me share my screen. Yeah. Is it visible?
Yes.
It's visible. Good. Yeah. Yeah. Once again, good morning, and thanks to IAFM for organizing this virtual conference. This is the third in the row, and I've been part with the last two ones, the last year and this year. It's my pleasure to talk on a topic which is very, 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 very strongly associated with the field of autism and other neurodevelopmental disorders. And I'll be talking about various aspects when it comes to the gut connection and what all can we do in our practice, and a little bit data from our own practice, what we have generated in the in 2023. We'll be just putting that also. We know that the GI tract is home to one of the most complex ecosystems, and it contains around 100 trillion microbes. We know that brain-gut microbiome access is a concept. It is not a new concept. It has been a concept which has been floating for years now, but somehow it has come to the limelight now. In the last few years, it's a concept that refers to the complex interaction between the central nervous system, the GI system, and the microorganism of the GI tract. And what is microbiota? Microbiota is a collective term for this microbial community, which includes bacteria, archaea, eukaryotes, and viruses. It will become more clear as I'll talk about it. So the term microbiota refers to an ecology community of commensals, symbiotic and pathogenic microorganisms that share a body space. There exists a symbiotic relationship between the host organism and the microbiota, which is codependent on each other. They're codependent on each other for survival. On the other hand, the term gut microbiome refers to the collection of microorganisms, their genome, their products that colonize the human intestine. And the gut has about 100 trillion microbes, bacteria, yeast, helminths, viruses, protozoa. And total microbial cells in the guts are more than the human cells in the body. The total weight of these gut microbes is around 1 to 2 kg, similar to the weight of the human brain. You see how it is. And the cumulative number of genes in the pool of microorganisms in the gut is larger than that in the human genome. So you see how important is the gut gene pool is. And gut microbiota has a key role in various direct inhibition of pathogens, production of cytokines, maintaining the structural integrity of the mucosal barriers, vitamin synthesis, and when production of short-chain fatty acids, production of short-chain fatty acids, as well as some gut hormones. And when there is disruption of this gut microbiota function, it leads to imbalances, giving rise to an inflammatory state and inflammatory-related diseases. Now, if you see in this diagram, how the brain and the gut, there's a bidirectional connection. How the entering nervous system and the vagus nerve is playing the whole thing. From the vagus nerve coming to the gut, and from the vagus nerve going to the central nervous system. How this is interconnected. And if you see that the round circle, the section of it, in the small part, a lot of things are happening. There's a gut lining is there. There the commensal bacteria are there. There the entering nerves are there. Immune cells are there, microbial metabolites are there, the various neurotransmitters which are getting synthesis, the bacteria, bifidobacterium, E. coli, neurotransmitters getting synthesis, tryptophan, clostridium. So all good, bad, everything is happening in this part of it. So you see a lot of things are happening parallelly. It's like multiple things are happening at one go. Now, so it is controlled by the autonomic nervous system, as I was mentioning, by the vagus nerve, which connects the CNS to the enteric nervous system. So it is there. It is there. And it is present in the mindric plexus and the submucosal plexus. And this plexus also contains the glial cells, mast cells, and sensory nerve endings. And all this communication is carried out by neurotransmitters like serotonin, GABA, some of which are produced in the gut, which requires healthy and variety-rich gut flora, good flora. So, and as you see in this diagram also, from that the main signaling pathway in the gut-brain axis, the neural pathways, endocrine signaling, immune signaling, HP axis, various neurotransmitters. Then we have a bacterial cell wall polysaccharides, LPS, and bacterial metabolites. All these things are helping in the communication. So a lot of things are happening. A lot of things are happening. So from hypothalamus to enteropituitary to adrenal gland, causing the metabolic effects. And the brain-gut microbiome axis is going on, and various messages are going simultaneously. I'll make it more clear as I go forward. Now, and we know all this is, and we all know the gut microbiome has been reported to be affected by multiple factors: age, region, food, gender, clinical comorbidity, and perinatal factors. Proteins, fats, digestible and non-digestible carbohydrates, probiotics, polyphenols, all can change the microbiome. And we know, and this, how do we get the gut microbiome, gut microbes, microbiota? Multiple ways: mode of delivery, vaginal birth versus cesarean section, gestational age, preterm versus full term, feeding mode, breastfeeding versus formula, environmental factors, and host genetic and epigenetic changes. Let's come to one by one, or to each factor. So when we talk about the mode of delivery, normal versus cesarean section, we know that the in utero colonization of the infant by the E. coli, Enterococcus, Staphylococcus epidermis occurs from the mother's gut through the bloodstream and the placenta. And that is what gets transmitted. So intestinal microbiota in vaginally delivered infants appear to be similar to the maternal vaginal microbiota. That's why the normal delivery really helps children to develop their immune system, and lactobacillus predominates. But on the other hand, the intestinal microbiota of the infant delivered by cesarean section is similar to the mother's skin and environment: lower bifidobacteria, bacteroides, and higher staphylococcus and clostridium. So you see, from there, the seeds are laid down. So normal delivery versus cesarean section. Preterm versus term. The prematurity is associated with a delay in the gut colonization with bacteria such as bacteroides and bifidobacterium species. And it's mostly dominated by Staphylococcus, Enterococcus, because of immature immune system. So if it's a normal delivery, if it's a term delivery, definitely the child's immune system is more activated. The immune system protects the child. Type of feeding: Bifidobacterium longum, by using oligosaccharides in the mother's milk, competes with pathogenic bacteria. That's why breastfeeding is so important. Maternal stress, again, can influence the development of the nervous system of the offspring by changing the composition of the vaginal microbiota. So that's why that's why a happy pregnancy always helps in the happy fetus and always helps in the happy baby. That's important, is that. Now, circadian rhythm also infects, and then some clock genes of the host bacteria such as PER, CRY, BMEL are found to regulate the human circadian rhythms. In the diet, short-chain fatty acids are products of microbiota metabolism, which are based on nutrients delivered with food consumed by the host, especially dietary fiber. So Mediterranean diet supplemented with fish oil can really feed the beneficial gut microbiota and improve the various symptomatology. Probiotics and antibiotics. Probiotics, in addition to restoring the intestinal balance, it reduces the risk of colonization of the intestine by the opportunistic pathogens. We know that prolonged and the frequent use of antibiotics can lead to gut dysbiosis. That's why as least as possible of antibiotic usages. And we have seen in children with autism spectrum disorder that there is a multiple use of antibiotics are there. Occupational environmental exposures. We all know heavy metals, pesticides. We are living in a toxic world. Heavy metals, pesticides, herbicides, polycyclic aromatic hydrocarbons, PAHs, or polychlorinated biphenyls in the environment can all modify the composition of the gut microbiota and thus affect the human health. Consumptions, drinking through the plastic bottles, drinking and consumptions using of aluminum foils, microwaves, all that can lead to modifying the composition of the gut microbiota. So the important is that simultaneously, alteration in the normal commensal gut microbiota termed as dysbiosis can again contribute to such symptomatology. The dysbiosis may be associated with the neurodevelopmental, neurodegenerative, and psychiatric conditions and modulation of the microbiota gut-brain axis by probiotic, prebiotic, or diet-induced preventive and therapeutic effects. So we know the dysbiosis, which we'll talk more and more, is an important contributory factor. So let's see how it happens. How it happens. So when the vagus nerve transmits signals from the gut microbiota, like Lactobacillus rhamnosus, which alters the central GABA receptor expression and results in reduced anxiety and improved cognition and communication. The immune signaling, very, very important. The disturbance in the intestinal microbiota, it damages the enterocytes. It weakens the tight connections between them. And once that happens, there's a leaky gut. And this translocates the gram-negative bacteria, which contains the most important LPS, lipopolysaccharide, and opioid-like substances from wheat and milk, causing the overactivation of the immune system. So you see how the damage is happening and how the whole cycle leads to the overactivation of the immune system. The immune system has to be activated, but not overactivated. And then the microbiota metabolites upregulate the T regulatory cell synthesis and secretion of the anti-inflammatory cytokines IL-10. Also, the oral consumption of Bifidobacterium infantis is associated with enhanced IL-10 expression in the peripheral blood. So all this immune signaling is happening, which contributes. So we have good and the bad thing happening. Endocrine signaling. The gut microbiota produces numerous substances like GABA, which alters the lactobacillus bifidobacterium, acetylcholine, lactobacillus, serotonin, E. coli, Candida, enterococcus, dopamine, bacillus, or noradrenaline bacillus saccharomyces. So these all are involved in communication with the intestinal microflora and the brain functioning. It also influences the level of metabolite of glutamate, which is involved in learning and memory. Also, I want to emphasize on HP axis how the stress raises the level of cortisol, which is implicated in gut dysbiosis. How the serotonin and tryptophan pathway, like 95% of the serotonin, is produced by the gut mucosal, enterochromaffin cells, and the ENS neurons. And the peripherally, the serotonin is involved in the regulation of the GI secretions, motility, and pain perceptions. In the brain, the 5-HT signaling pathways are implicated in regulating the mood and the cognitions. And the bifidobacterium, which promotes the 5-HT biosynthesis from enterochromaffin cells, and it plays an important role. Now coming to the LPS in Clostridium, Proteus, and Enterobacteriaceae produces ammonia, a potent neurotoxin. And Clostridium can reduce the secretion of dopamine beta hydroxylase, which results in the deficiency of norepinephrine and excess of dopamine. That's why we need to address Clostridium, otherwise it impacts the dopamine. And short-chain fatty acids, it produced by beneficial bacteria. They are produced by beneficial bacteria like butyrate. It nourishes the colonocytes. It inhibits the pathogenic bacteria, such as E. coli, maintains the correct pH. And propionate produced by pathogenic bacteria like Clostridium, it reduces the short chain fatty acids, which is implicated in autism spectrum disorder. So you see how these multiple things are happening. So putting all of them together, the immune response against regulation of gastrointestinal functioning. Once that happens, then what happens? There's accumulation of all this happening, and then there's a leaky gut which is happening. Once the leaky gut is happening, opening of the tight junctions happening up, and the entry of peptides, entry of toxins into the circulation into the brain. And then they reach to the brain and they bind to the opioid receptors, and they cause the neurotoxin and the inflammation of the brain, and that leads to the brain dysfunction. So the exact pathogenesis we are yet poorly understood. Nevertheless, studies have highlighted the interaction. And a lot of evidence is coming up that this manipulation of the gut microbiome could modulate and the neurotransmitter levels, neuroinflammation, and the brain functions. Now, as we come more and more to the autism spectrum disorder, the frequency of the GI symptoms in children with autism ranges from 9 to 84%, compared with 9 to 37% for children without ASD. So you see in your practice, when you see that the GI symptoms are so common. And the most common symptoms include flatulence, constipation, bloating, abdominal pain, diarrhea, burping, belching, and gastroesophageal reflux symptoms. So these symptoms are very common. So a lot of times when they have behavioral problems, we can correlate their GI symptoms with their behavior symptoms. Aggression, agitation, sleep problem, getting up in the middle of the night, sensory issues, all those can be correlated to the GI symptoms. You improve the GI symptom, the behavior improves. And also, absent and the delayed acquisition of the bowel training, secondary to difficulty with sensory stimuli, sensory processing and motor problems may lead to altered GI motility and defecation physiology. That's what. So it's a lot of times the bowel training, it does not happen because the gut issues are there. Once you address the gut issues, once you work on the GI motility, you find gradually the bowel training happens. They often present, the children who have GI symptoms, they often present with more severity compared to the 8 autism children with without GI symptoms. And the behavioral characteristics with a hypothesis to be expression of GI problems in children with ASD includes facial grimacing, teeth gritting, excessive chewing, posturing, one of the most common, posturing, pressing over the abdomen, irritability, oppositional behavior, and sleep disturbances. So usually these are the behaviors, and then you correlate with the GI symptoms. Now, I just want to highlight to this slide, which talks about composition of the gut microbiota in different neurodevelopment disorders. And if you see the various studies which have come from the Western world, which talks about that how children with autism, the gut compositions, like in the bacteroid format, in this slide, and this one, how the bacteroids, formatides, and the Clostridium is very predominant in this. If you see in this slide, is that the bifidobacteria and lactobacillus. In these some of the slides, the Clostridium was prominent. In some of these slides, if you see, again, the bifidobacterium was prominent. Here, the Votella was prominent. See, and the metabolites was short-chain fatty acids like acetate, propionate, and valerate. The fecal butyric, isobutyric, fecal ammonia, calperic acid. Now, if you sometimes club all the symptoms together, you will see some of the symptoms which talks about is gut dysbiosis, like insatiable appetite despite poor weight gain, aggression, worsening at the full moon, picking, biting, licking, anal scratching, teeth grinding, restlessness. All this indicates towards parasites. Takes OC symptoms, they more go through the strep infection. Inappropriate laughter, sugar craving, frequent diaper rash, frequent urination, history of frequent antibiotics, night awakening. They mostly go towards your yeast issues. Aggression, temper issues, agitation, irritable, very foul stools, mucosal stools, severe diarrhea following antibiotics. They more and more go towards Clostridium. Squinting, divergent gaze, cold sores, warts, history of developmental regression, they more and more go towards viral. Flatulence, bloating, constipation, abdominal, intestinal diarrhea, burping, belching, gastro, they all goes towards SIBO, small intestinal bacterial overgrowth. So you see how we can club the symptoms together and pick up that without even getting the testing, we can identify that these symptomatology indicates there is a particular type of gut dysbiosis which is happening in this particular child, and we can address it that even without doing expensive investigations also, because this can help us a lot of times the affordability, the cost-effective interventions also have to be into consideration. Now, coming to the intervention. Now, up till now, I've given just a brief overview about the gut microbiota, the various factors which can contribute to lead to the various symptomatology of autism spectrum disorder. What are various various manifestations which can present to clinician, a behavior manifestation which we can identify can be like a gut dysbiosis. Now, coming to the what are the various interventions which can do, one of the most common intervention which is recommended is the dietary interventions. And we have a generalized diet protocol and the specialized diet protocols. And many reports shows favorable results in the core of peripheral symptoms of autism when we follow like a GFCF diet, the communication, language, social interaction, stereotypy behavior, hyperactivity, GI symptoms. So they are becoming a popular choice. So coming to the generalized diet protocol, in generalized, we have to avoid the refined processed foods, artificial sweeteners, preservative, additives, sodas, deep fried food, canned, sugar, caffeine, refined. So this is something, a general recommendation to include organic and the fresh foods. I always recommend the parents, if they can go to the farmers market, if they can collaborate with the farmers, the fibers, protein, seafood, poultry, meat, beans, fermented foods, very, very important, good fats, oils, healthy drinks, all this has to be included in a generalized diet protocol. Then we have specialized diets. We have gluten-free casein, we have gluten-free diet, we have casein-free diet, we have soya-free to eliminate gluten, casein, soya. Correct. Let me come to this one. Where we have gluten-free, casein-free, we eliminate wheat and the milk products. And we know that the casein and gluten, they may be incompletely digested to peptide and may enter the bloodstream from the intestine, while an abnormally permeable intestine lining, which is called as leaky gut. And once they cross it, these partially digested protein peptides, it can potentially cross the blood-brain barrier and negatively affect the brain function, almost like an opioid addiction. So there is craving for these opioid food sources and limited appetite for other foods. You will find them, they're very, 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 very picky eater. They will have a very, very strong craving for only milk, or they just will be drinking only milk throughout the day. And once you stop them, the improvement in certain behaviors of autism, such as social isolation, communication, overall behavior, in almost 2/3 of cases, this is 69% of them show improvement. When we talk of STD diet and body ecology diet, so we have to eliminate foods that feed yeast overgrowth, such as sugars, yeast, starches, fruit juices, refined grains, and processed meats. The logic is that, the rationale is that these diets are helpful for individuals who have chronic intestinal yeast overgrowth that is not adequately treated by probiotics and antifungal medication. So after giving multiple rounds of probiotics, antifungal rounds, then also they don't improve, the yeast symptoms don't improve, and the inflammation continues. So around 72% of children, they show improvement. Plain gold diet, phenols and salicylates, containing foods such as apples, red grapes, tomatoes, artificial food additives. So the enzyme phenol sulfotransferase, PST, it uses sulfate to metabolize phenols. So when PST and other sulfate are deficient, phenols fail to metabolize well and reaction occurs. And the symptoms are like red cheeks, ears, aggression, hyperactivity, night sweats, and craving of phenols and salicylate. Once you remove them, you find improvement in aggression and hyperactivity. Low oxalate diet. This is one thing which I've seen a lot of improvement in behavior when you do low oxalate diet. So high oxalate foods such as peanuts, okra, spinach, nuts, and they can cause digestive pain when absorbed by leaky gut, can elicit symptomatic pains and kidney stones. They keep on pressing their body parts, corner of the eyes, chin, and the genitalia. So, and once you remove them, there's improvement in the pain and the tactile sensitivities. Other interventions like microbiome therapeutics, like microbial therapies are widely popularized for using beneficial microbes naturally found in the human body. It has undergone several advancements like genetic engineering. Now, one of them is additive therapies. We'll talk about multiple ones. So additive therapies, where a cocktail of beneficial microbes is extracted from the healthy human body and introduced in the fecal microbial FMT, fecal microbial transplantation, or through the consumption of the probiotics. Now, we know that FDA USA awarded the fast track status to FMT therapy for children with ASD, apparently considering its influence on ASD symptoms and progression via multiple gut-mediated immune, endocrine, and direct neural pathways. So it has been found that FMT not only relieves gastrointestinal symptoms, but also the neurobehavioral symptoms. With the Kang et al., they reported that in a trial of 18 young patients, that both gastrointestinal and the behavioral ASD symptoms improved, and this improvement persisted up to 24 months after discontinuation. And what they found, the intestinal symptoms often improved after FMT. And that's why It was a fast track which was given up, and a lot of studies are coming up, and a lot of people are working, and the promising results coming up. Coming to the probiotics, we know the probiotics, how do they act, and we have a lot of talks on the probiotics. The probiotics act by restoring the microbial balance, inhibiting the growth of pathogenic microorganisms, production of short-chain fatty acids, supporting the gut barrier integrity, and immunomodulation. Lactobacillus and bifidobacterium strains have been proven to have anti-inflammatory properties by reducing the level of inflammatory cytokines such as IL-2, interferon gamma, IL-4, IL-13, and IL-17A, and increasing the level of anti-inflammatory cytokines IL-10. So they have anti-inflammatory properties also, and they increase the level of anti-inflammatory cytokine IL-10. Multi-strain probiotics may imply, there's always a controversy, single strain or multi-strain. So multi-strain probiotics also may imply more chance of success. It can mean a broader spectrum of efficacy, and there's often the hope that there are potentially synergistic effect. And however, structured research in this regard needs to be more conducted in this. So is there, and some of this, it's a review paper which came up with one of the authors from India, Himani Khanna, and in which she has reviewed, and overall she found that Lactobacillus paracasi, 37, Lactobacillus planetarium, Lactobacillus reuteri, and Bifidobacterium longum were the most promising strains of probiotic for treating children with neurodevelopmental disorders such as ASD and ADHD. We also know Lactobacillus reuteri works through oxytocin. We know that Lactobacillus rhamnosus, again promising, which works through GABA, and Bifidobacterium longum works through BDNF. So in that way, all these probiotics have a mechanism to work on and has been found to be effective in children with ASD and also in other neurodevelopmental disorders. Again, subtracted therapy, where they engineered the bacteriocins and bacteriophages that exert promising antimicrobial activity, are used to target pathogens in the gut without causing harm to other microbes in the ecosystem. And so they are the peptides, bacteriocins are peptides, and the bacteriophages are viruses. And more research are required, and before we can more human studies are required before we can use it. And it looks to be very promising. And again, modulating therapies, like restoring a healthy balance in the gut microbiome by changing the diet, exercise, antibiotics. Various speakers in the last 2 days have talked about it, and we'll be talking about which favors the colonization of the beneficial microbiota over the pathogens. So there's no second thought that modulated therapies are important. A healthy diet is very important. We have already talked about, and exercise is also very, very important, how they can enrich the microbial diversity, leading to increase in the bacteriocytes and firmicides ratios, contributing to the reduction of metabolic disorders, and modulating the production of bacterial metabolites like short-chain fatty acids. And we have enzymes and antimicrobial protocols are there, and digestive enzymes are there, who have gastrointestinal disturbance resulting in reduced ability to digest proteins and thereby availability of amino acids. So one of my favorite questions which I ask them, how's the poo? And if the poo has indigested food, then definitely your digestive enzymes really helps. So along with the probiotics, using of digestive enzyme, and as I was talking about the various symptomatology, if they have yeast issues, parasite issues, strep issues, viruses issues, using tailor-made antimicrobial protocols, antifungal, antiparasites, antiviral, it also helps in specific signs, symptoms associated with some symptoms of ASD. Now, there are various other protocols like Nemchuk protocol I've used in a lot of children with promising results. It aims to rehabilitate the brain by targeting the autonomic dysfunction and by rebalancing the intestinal microflora. It focuses to reactivate the brain's neuronal pruning by lowering the inflammation. So you decrease the gut inflammation and the brain inflammation, promoting the stem cell growth and recreating a natural chemical environment that child brain needs to correctly control cells, organs, and body. It uses virgin oil for cooking and introducing inulin, omega-3 fatty acids, and rifampicin gradually, and vagus nerve stimulation in the step-by-step procedures. So I'm almost in the end part of my presentation. This we tried for this conference a little bit, that around 470 children in the last 8 months of diagnosed with ASD. In a consecutive sample, we saw in 50% of children, we saw having constipation, the chronic constipation, around less than 1% had diarrhea and the loose stools, and around 50%, they had minor gut issues and regular gut. It was very difficult for the parent. That sometimes we had little difficulty, sometimes it's normal, but yes, you can see how in this, how the gut is very, very strongly involved in children with autism spectrum disorder. So before I end, there's few challenges are there in the future perspective. A lot of studies have been conducted in experimental animal models, and some have small sample sizes. Some of them lack randomization and control groups, so we need more and more studies. But there are a lot of increasing number of articles which have been coming in science media, which are featuring the success of microbiome research and its promising therapeutic regime. Further research should be conducted to determine that how the different population of ASD patients across various geographical locations and how they can help them. And I'm sure in the next couple of years, we'll have more FDA-approved pills, probiotics, and metabolites, which might be commercially available to establish a suitable bacterial composition to treat and regulate ASD. So the takeaway points are that a lot of children with ASD have impaired GI functions. Clinicians working with such children increasingly assume a link between ASD and gut dysfunction. Several factors lead to ASD which have been identified, mainly early colonization on infant health development, effect of early microbiota dysbiosis in the gestation period, motor delivery, uncontrolled use of antibiotics. Gut microbiota interacts with CNS by influencing the neuroendocrine immune system. Controlled manipulation in the microbiome in the form of dietary modification, probiotic, digestive enzymes, FMT is a promising area, and further studies are required in Indian population, especially to enhance the understanding of correlation between gut-brain axis and help to formulate management guidelines. Thank you very much. And these are some of the references. And thank you, IAFM, for giving me this opportunity. Thank you very much.
Thank you so much, sir, for bringing out this connection between gut and autism in such a simple way. There are a lot of questions in the chat which I would have taken, but now since we are we are short of time, I'll request you to please stay back and answer the questions in the chat. And we will take more questions in the panel discussion later. Thank you so much, sir.
I'll try to answer as much as possible in the chat.
Sure, sir.
And the rest we can talk in the panel discussion at 12 o'clock.
Absolutely.
Yeah,
Yes.
thank you very much.
Thank you.